Greetings and welcome to NSCS 200, the Fundamentals of Neuroscience and Cognitive Science. My name is Allison Peel, or Dr. Allison Peel if you like, and I'll be your guide along this, what is hopefully a very exciting journey. So briefly about myself, I received my PhD from the University of Florida College of Medicine. My graduate work was in looking at the feasibility of using viruses to express genes in mammalian nervous systems. So it's a field called gene therapy. And we were the first to show that you could actually express the jellyfish green fluorescent protein in a mouse model in vivo using an adeno-associated viral vector. That particular vector system is currently being used in clinical trials in humans for a number of different disorders. After receiving my PhD, I was invited to do a postdoctoral fellowship at the Buck Institute for Research in Aging in Marin County, California. While at the Buck, I looked at the molecular underpinnings of neurodegenerative diseases with a focus on Alzheimer's and Huntington's diseases. Now this is a video that I've recently updated, amended a few things, largely because all of the information is relevant and still timely, but needed a little bit of editing and additions. So if you come across something that doesn't make sense or looks like it may have been edited in and you have questions about it, feel free to contact me with those questions. How and why we study the nervous system. So when I was an undergraduate and I found out about the field of neuroscience, that we could actually study the brain and find out how it works in a very real way, I wondered why anyone would study anything else. Truly, study the nervous system. The nervous system is how we are in the world. We are simply sense organs for everything that's going on around us. We sense it, we take it in, we process it, and we act upon it. So the nervous system pretty much is how we are in the world. The how of it is a little bit different, and we will be studying that. We will be exploring that in a number of sessions. So when we look at the evolution of the nervous system, the brain itself and the nervous system evolved. It was started to evolve from organisms that have had nerve cords about 500 or 600 million years ago. And from those very, very simple nerve cords, everything else subsequent to that, leading to this very complex human brain, has simply been layered on top. It's kind of like starting with a very small house and then adding a room here, a floor here, a garage. So we've just kind of added on. There's a researcher at Johns Hopkins University. Let me have you take a look at this here. David Linden, and he's actually written a very good book about the evolution of the brain, talking about or postulating that it's pretty inefficient. The way it's put together is pretty inefficient, largely because it is a function of evolution. It is a function of just adding stuff on, tweaking here, changing there. That if you were going to design a human brain from the bottom up, you probably would do that a lot differently and a lot more efficiently. It's a great read. It's a relatively recent book. Again, David Linden, The Accidental Mind. So if you have some time and interest, and I hope you have the interest because you're in this class, then you might want to take a look at that. Okay, so back to how and why we study the nervous system. Evolution actually did us a favor by layering on, because it allows us to study the nervous system in very simple organisms. We evolved from simple organisms, and so a lot of those structures and those basic processes are the same across species and from very simple to very complex organisms. So we use a number of models, simple models, to study things like the action potential, synaptic transmission. The basic processes are the same in Drosophila, which is the fruit fly, C. elegans, which is a microscopic worm, both of which are used to study the nervous system, all the way through more complex organisms.
This even applies to sensory and motor circuits, learning, and looking at, at a molecular level of what's going on. We're able to use these simple organisms. There's a researcher, his name is Eric Kandel, one of the brilliant pioneers in neuroscience, that used the sea slug, Aplysia, to learn some things about reflexes and how nerves actually work. That translated to more complex models. While simple systems will take us so far, and they do, and we've learned a lot from them, we really need to, in order to understand our brains, to look at more complex models, particularly mammalian models. We use a lot of rats and mice in neuroscience research. We did so in the Gene Therapy Center at Florida, looking at gene therapy approaches to neurodegenerative disease. And from those successful attempts, other labs have taken that approach and used gene therapy to look at functional studies of the nervous system. Even rat and mouse models limit us somewhat, and some things you don't understand until you move into a more, into a primate model, for example, which then leads us into a lot of ethical concerns. There's been a lot of press about using primates for research, and rightfully so. It's very, very difficult to get primate research passed by an internal review board, but that's as close as we're going to get without studying on actual humans to understand the human brain and its potential. So you need multiple layers of analysis to study the brain. All of these factor into all of these, lead us, give us good information, and put together kind of give us a whole picture. So the molecular level, we're able to study DNA, we're able to study RNA transcripts inside single cells. We're able to look at it at that finite a level. We also look at it at cellular levels. We look at enzyme reactions in individual cells and how that contributes to thinking, to memory, to disease. We look at systems level, not only the brain as it's a system in your own skull or in your own body, but also as the brain's a system in a larger world. We need to study how the brain affects behavior, and how behavior can affect the brain. So it works. It's a two-way street. It works both ways. We also need to take a look at how it all puts together into a cognitive approach to the nervous system. So let's look at cells of the nervous system. So you will hear consistently from a variety of sources that there are two major cell types of the central nervous, or the nervous system. There are neurons, as shown here, and then people say there are glia, as shown here. And glia is a Greek word for glue. So for most of the time that we've studied neuroscience, it was thought that the glia cells didn't do a whole lot more than simply provide support for the neurons, which were the main event and still are the main event. But that's not entirely true, and so we kind of have to revisit this neurons and glia. I like to think that, and again, you're to understand that there are two types, neurons and glia, but really, if you look at the central nervous system, you see that there are really four major cell types. There are neurons, okay, as you see there. Let's move these aside. And then there are three major types of non-neuronal cells, and you will be expected to know this because they have very different functions and very important functions. So first you have your astrocytes, shown here and here. Astrocyte end feet provide structure for there. They really comprise the blood-brain barrier. Then you have oligodendrocytes. Other people will refer to them as oligodendroid glia, and we'll talk about their role in sheathing nerve axons to improve the speed of nerve conduction. We'll talk about that at great length. In the peripheral nervous system, the cells that do that, and we'll talk about them as well, are Schwann cells, and they do differ from oligodendrocytes or oligodendroglia. Then finally, the fourth major cell type in the nervous system that comprise the nervous system are microglia, and they are the immune cells of the nervous system. Now let's take a closer look at what's considered a prototypical neuron. You will see these pictures of these. My drawings reflect this. So what you have here is, let me go back to my marker, is you have the soma or cell body. You would be expected to know the word soma. You have dendrites, which are tree-like structures that come off the neuronal cell body or soma. You have the axon, which is this long thing here that branches out into axon terminals. Connecting the soma, the cell body, to the axon is what's referred to as the axon hillock there. Okay, so it's this kind of structure here. It's not quite cell body, it's not quite axon. Used to think that the action potential was generated at the axon hillock. In fact, when I first started the neuroscience program at Florida, that's what we were taught. But since that time, we've come to understand that that's not quite true. That predominantly the action potential is generated near that area, adjacent to that area, but not exclusively. And then what you see in many neurons, but certainly not all neurons, are these myelin sheaths here. And again, I think I mentioned oligodendrocytes in the last slide, and they actually produce the myelin sheath. It's a fatty sheet. Inside, and we'll go at these in greater length in a few minutes, are the organelles, the structures inside the soma, inside the cell body, that are the machinery for the cell. You have the Golgi apparatus, the nucleus, the mitochondria, the endoplasmic reticulum, and the ribosomes, all of which you should have been exposed to in previous cell biology classes. There are many different particular neuronal cell types. There are dopaminergic neurons, serotonergic neurons, there are Purkinje cells. There are many different types of subsets of neurons with a number of different subfunctions. However, I would be bold enough to suggest that the primary function of neurons in the nervous system is to process and transmit information. And I didn't come up with that. If you look, you do a Google search for primary function of neurons, you will see just that, is to process and transmit information. You'll have people say that no, there's more to it, but when you boil it down to one sentence, that would be what I would want you to understand. The prototypical neuron, much like many other cells, contains the same type of equipment. You have your cytosol, or otherwise known as cytoplasm. It's a watery or aqueous solution, and inside there's the organelles. Organelles are membrane-bound, membrane-enclosed structures within the soma. It also includes nutrients and proteins. You have filament proteins, neurofilaments, that are involved in structural activities, transport. You have dissolved amino acids and sugars and fatty acids, a number of things inside that cell body. Let's take a look. Let's take a closer look. There we go. So you have mitochondrion, which is here. Nucleus, obviously, more towards the center. You have the neuronal membrane, which we'll talk about in a minute. The rough endoplasmic reticulum. You have ribosomes on the rough endoplasmic reticulum, and you have free-floating ribosomes. And you have the smooth endoplasmic reticulum as well. A couple things not shown in this slide is you also have, in addition to the neuronal membrane here. You have a nuclear membrane here. So the nucleus also contains aqueous nucleoplasm that is the home to the chromatin. Chromatin, as you know, are DNA and proteins kind of tightly bound together. You also have inside the nucleus a nucleolus, which is comprised of ribosomal. Actually, it's the site of ribosomal RNA synthesis. Is where ribosomal RNA is made, and the formation of ribosomes. So here you have the nucleoplasm, which contains the chromosomes and nucleolus, and also substances like nucleotides and enzymes that are required for DNA synthesis. The nucleolus, where ribosome biogenesis occurs, so it's where ribosomes are made. The nuclear membrane, right here, composed of a lipid bilayer, much like the cell membrane, and the nuclear pores, as you see here, these things that control the movement of molecules between the nucleus and the cytoplasm. So here it is in kind of a simplified version up top here, where you have the inside of the cell is here, the outside of the cell is here, and you have your lipid bilayer here. So this is one layer here, and this is the other layer here that forms the neuronal membrane. And that's a very simplified view, so you're looking at this down here. So let's kind of delve into a picture that I thought was a little bit more useful. Here we have intrinsic or integral proteins, which go from one surface of the bilayer membrane to the other. They span the membrane. You see these commonly in ion channels. And you have peripheral or extrinsic proteins, such as this, that attach transiently just to one surface or the other. Then we have these bipolar phospholipid molecules here. We have a bilayer of them. So on the outer surface, you have these hydrophilic heads here, so they span the entire length of the membrane. And then in the other phospholipid molecule here, philic meaning to like something, and hydro, of course, water or aqueous substances. So these outer surfaces like a watery companion. So it makes sense that you would see these separating the outside of the cell and the inside of the cell, which is also filled with aqueous solution. The other part of these phospholipid molecules are the hydrophobic portions here. So hydrophobic, hydro, water, phobic, fear. So to fear or to turn away from watery substances. So these naturally turn inward to face each other. The composition then of this, of this bilayer then that keeps watery substances contained sets the stage for the electrochemical properties of the neuronal membrane that allow for action potentials and everything that goes on subsequently in the brain. We'll be delving into a lot of that in more depth later. And I'm not going to go into gene expression too much at this point. It takes place in the nucleus. As you know, you have the unwinding of DNA. Let me point that out over here. So DNA unwinds, DNA is transcribed into RNA via RNA polymerase, and the RNAs, the mRNA transcripts then are exported out of the nucleus and into the cytoplasm. Transcription factors bind RNA polymerase to the promoter region of the gene and allow for the beginning of RNA transcription. That transcription ends at the termination or end sequence. The RNA then undergoes further modification called splicing, whereby regions are spliced out of the RNA and the exons or coding regions are spliced together. The edited mRNA then leaves the nucleus via nuclear pores and moves to sites of protein translation where the mRNA is translated into proteins. We will go into this in a bit much more detail further on in the class.
But briefly, here's a small video clip of what's going on.
This slide shows protein synthesis as it takes place at the ribosome. In this case, the pictures are looking at free ribosomes here. They look free anyway. But it also takes place at ribosomes on rough endoplasmic reticulum. The thing I'm going to kind of take issue with in this slide is it says here the soma, which as you know are the cell body, and some major dendrites are major sites for protein synthesis. However, there's pretty good evidence now that protein synthesis occurs in the dendrites, in the axons, and in the axon terminals. The smooth endoplasmic reticulum and the Golgi apparatus are sites where proteins are prepared and sorted for delivery to different cell regions. Particularly, or specifically, the smooth endoplasmic reticulum is associated with the nucleus and delivers proteins to the Golgi apparatus or Golgi body to be modified, sorted, and packaged into macromolecules that then are secreted from the cell. The mitochondrion, or plural mitochondria, are particularly important for the brain. The human brain uses about 20 to 25% of its basal metabolic rate for the brain, and so the metabolism of glucose and turning glucose into energy or food into energy is crucial in the brain. So these puppies are really, really important. What's kind of neat about, and the Krebs cycle, again, which you should have all been exposed to already, is the production of ATP. Let me see, that's down here. Okay. So you have dietary sources of energy that go into the synthesis of pyruvic acid that then go into the mitochondria for the production of ATP and carbon dioxide. So what's interesting about the mitochondria and kind of consistent with what we see in the brain is this folding of the inner. So the mitochondria has two membranes, the outer membrane and the inner membrane. And the inner membrane is where all the activity, the ATP synthesis, takes place. And the purpose of the folding is very efficient, much like the brain. So the brain has gyri and sulci. Those are hills and valleys that you see, the convolutions in the brain. And what those do in the brain, and the purpose that these convolutions provide in the mitochondria, is an increase in surface area. So it's very, very efficient. It's not so much in terms of a brain or mitochondria, it's not so much how big, but rather the surface area that's generated by these convoluted areas. Another interesting feature of mitochondria are the hypotheses, or the hypotheses about how they originated. The most favored one is what we call the endosymbiotic hypothesis. And endosymbiotic hypothesis essentially says that at one time mitochondria were prokaryotes that existed independently of eukaryotic cells, and they were capable of carrying out a lot of the production of energy that your eukaryotic cells were not able to do. And so somehow they formed a symbiotic relationship, or some people would say symbiotic relationship, and fused together, allowing eukaryotic cells now to kind of jump into a whole different form of evolution. The support from this comes from the fact that the mitochondrial membrane is actually very similar to the bacterial membrane, and so this is a hypothesis that seems to have gained a great deal of favor. So I want you to kind of take a look at that. Maybe do a search. Just Google it. Google bacteria and endosymbiosis, and kind of take a look at that, because it's very, very interesting, especially given how important mitochondria are in the brain. Another important feature of neurons, a particularly important feature of neurons, are the cytoskeletal proteins. You have microtubules, comprised of the tubulin molecule, and this is really important in how that's regulated by tau, a protein called tau, which is involved in the pathophysiology of Alzheimer's disease, and we'll talk about that certainly later. You have neurofilament and microfilament proteins that are involved in any number of things: growing growth cones, the lengthening of axons, the transport of molecules down the axon and throughout the cell. Cytoskeleton protein is very, very important in neurons. You have two ways that proteins are transported in the axon. You have anterograde from the cell body to the axon terminal, so anterograde transport, and you have retrograde transport, where proteins are perhaps taken up at the axonal cleft, at the synaptic cleft, and taken back up into the axon and retrogradely transported to the soma. So what about the axon? It's kind of comprised of three parts: the axon hillock that we talked about, and that is this part right there. See that axon hillock? Then you have axon collaterals, and we didn't mention those, and I'm not sure I mentioned the nodes of Ranvier, but you have the axon that's myelinated via Schwann cells or oligodendroglia, and at the nodes of Ranvier, which are those small spaces between the myelinated areas, so that's a node of Ranvier, R-A-N-V-I-E-R. At the node of Ranvier, you will sometimes see axon collaterals branch out. Oftentimes, the function of these axon collaterals are to feed back onto the cell body and to modulate what's going on at the level of the axon. And then finally, you have the axon terminal, or end of the axon. The axon terminal is the business end of the axon, where all the important work takes place, or most of the important work takes place, where synaptic transmission takes place, where electricity is turned into neurochemistry. It is the main event here. So let's take a look here. Here you see, this as well, sorry about that. Okay, so here you see the synapse, this is the presynaptic terminal here. So this is the axon terminal there. And you have the postsynaptic, in this case a dendrite. It's a postsynaptic neuron, but it's the dendritic arm of the neuron. So here you have the synapse. It's where the axon terminal comes to meet the postsynaptic dendrite. At the very end of the synaptic terminal, at the end of the axon terminal, are synaptic vesicles. These vesicles contain the neurotransmitters, neurochemicals that are released following an action potential. So essentially the vesicle, and we will talk about this further when we talk about synaptic transmission, these vesicles will bind with the membrane at the end of the nerve terminal and open up, releasing the chemicals into the synaptic cleft, which are then taken up by their very specific receptors. So here you see here the synaptic cleft, which is simply that space in between the axon terminal and the postsynaptic cell. And way at the other end from the axon terminal, so you see the axon, whoops, sorry about that. You see the axon coming down here and maybe making a connection here, so this is at the terminal. Here you have the soma, the cell body, and you have the dendrites, and they're beautiful arborizations, tree-like, branch-like structures. This is where you have a lot of receptors, so that when an axon then terminates here, it delivers its package of goods to these dendrites. And you have any number of inputs coming in at any one time on any one dendrite. What makes it very interesting, because you see kind of a pattern evolving here, is the presence of dendritic spines, which they don't show here. So dendritic spines are little dots, they're not dots, they're little tiny structures. There's actually a neck and then kind of a small bulbous structure there. And what that does, these little structures, and I'll show you how that works in a real dendritic spine, is it also, much like the gyri and the sulci and the convolutions of the mitochondrial membrane, what it does is it increases the surface area that connections can be made. So, let's see, here you have the dendrite right here. Dendrite, this long thing here. Here you have the spine neck, which makes sense, so spine neck. And then you have the head of the spine here. Very important structures. Again, the function of the dendritic spine is to increase attachment sites or connection sites for presynaptic connections to be made. A key feature of these spines is that they're very, what we would call plastic, in that they change their shape, they change their location as a function of actin remodeling inside the dendritic spine. And because they change their shape based on the activity that's going on from the neural connections, from the axonal connections to them, and the release of neurochemicals. And so they're believed to play a major role in memory formation and neuroplasticity. Astrocytes have a fair number of functions, the main ones of which I think I've listed here, and it would do you well to know these. The first one listed here, and again they're listed in no particular order of importance, is that astrocytes synthesize neurotrophic factors. In particular, the one that they're best known for is glial cell-derived neurotrophic factor, or GDNF. Glial cell-derived neurotrophic factor has been shown in vitro and in vivo to rescue both dopaminergic neurons and motor neurons. And in fact, one of the groups at the Gene Therapy Center collaborated with us to show successful rescue of dopaminergic neurons in a Parkinson's model using viral vectors that were expressing GDNF. Next, they secrete extracellular matrix proteins. So extracellular matrix is a substance that the cells, the neurons, and glia move around on, they develop on, provides growth and nutrition. So astrocytes play an important part in producing extracellular matrix. Astrocytes also function to regulate the blood-brain barrier. The endothelial cells of the blood-brain barrier of the blood vessels actually, with their tight junctions and the basal lamina, the membrane that they lay down, really comprise the core of the blood-brain barrier. But it looks like, and it was thought that astrocytes played a more important part, but it looks like astrocytes may be involved in the regulation of these endothelial cells, and how the endothelial cells develop. Astrocytes play an important part in the immune response. In some way, it's almost detrimental. In working in a spinal cord lab, we would see where a spinal cord injury, in inflammation, astrocytes rush into the site of the injury in response to the chemicals that are released at that point. They start to proliferate, they start to divide, they multiply, they proliferate, and they form a barrier around that injury, which is a good thing, so that keeps the injury from spreading. The bad thing is they create what we know as a glial scar. Glial scar. And that's what keeps regeneration from happening in the central nervous system, is the formation of this glial scar, so that neurons that actually are able to stay alive are not able to send projections back out to where they were through this glial scar formation. Astrocytes store and release glycogen, which is an important source of energy for the brain. They regulate ion concentration in the extracellular space. Specifically, astrocytes have a high concentration of potassium channels, and when potassium is released into the extracellular space, let's say in the case of an action potential, astrocytes serve as a potassium sink, soaking up excess potassium so as not to continue to depolarize the cell. Astrocytes are also known to clear and metabolize neurotransmitters that are in the synaptic cleft. This is a really good thing. For example, if you have too much glutamate, that's an excitatory neurotransmitter. Too much glutamate can cause excitotoxicity, or the sickness and death of neurons by the presence of too much glutamate. And so the astrocytes perform an important function by breaking down, metabolizing the excess glutamate and other transmitters, particularly the amine transmitters from the synaptic cleft.
Now, astrocytes come in two flavors. Fibrous astrocytes, which are found in the white matter tracts. Those are the axon tracts in the brain and spinal cord. And protoplasmic astrocytes, which are found primarily in the gray matter. Now, there's a third type of glial cell that occurs predominantly in the developing brain, but is also found in the adult brain in certain areas, called the radial glia cell. Okay, and that's what these long things are here. So they're actually progenitor cells that can develop into neurons, astrocytes, and oligodendrocytes. But in early development, these cells send processes out to the surface of the brain, and those are the cells along which baby neurons migrate to form the developing brain.
So here we have an opportunity actually to kill two birds with one stone. What you're seeing is the cover of the journal Practical Neurology. It's a scanning electron micrograph, so it's looking in really closely at brain tissue, and it's actually looking at brain tissue from multiple sclerosis brain. And so multiple sclerosis is a disease in which the body's immune cells, in particular the central nervous system immune cells, start to attack the brain's own cells. And so what you see here are, you have oligodendroglia, you have microglia, which are the immune cells of the central nervous system, and they're attacking, as you see here, not very well, you can see them here, the oligodendrocytes. So now, microglia normally ingest, as you see here, cell debris, and bacteria as a part of the body's immune response. What they're doing here, unfortunately, is they're attacking the oligodendrocytes. As you've heard me mention before, oligodendrocytes are the cells that wrap themselves around axons to improve nerve conduction. So they increase the speed of nerve conduction in the white matter tracts. So loss of the oligodendrocytes leads to demyelination. You lose the fatty sheath around those axons and you lose nerve conduction. So what you end up with the loss of the oligodendrocytes, the loss of the myelin sheath, is deficits in vision, in speech, and in movement, as are characterized by multiple sclerosis.
I'm also uploading a couple articles, one on microglia and one on oligodendrocytes, a couple review articles. They seem rather lengthy. I think I have highlighted the really important features. What I would like you to do most is understand the varied functions that these different cells have in the nervous system. Let's take a look at cells that myelinate axons. So up here you have oligodendrocytes. Oligodendrocytes are the myelinating cells of the central nervous system. Schwann cells down here are the myelinating cells of the peripheral nervous system. One primary difference between the two of them is that one oligodendrocyte can myelinate several axons. You see here the axon in the center with the myelin sheath wrapped around it. In the peripheral nervous system, one Schwann cell myelinates an axon. So here you have 3 Schwann cells, this one's shown in cross-section, that are myelinating an axon. In between the axons, in both cases, you have unmyelinated segments that we call the nodes of Ranvier. These unmyelinated segments here, which we'll get into in much greater detail later, are important for the propagation of an electrical signal down the axon. So again, understand, oligodendrocytes myelinate axons in the central nervous system. Schwann cells myelinate axons in the peripheral nervous system. Myelin is made up of proteins and fatty substances that provide an insulating layer around the axon. So as a postscript, I left these out of the neuronal lecture and they should have been included. So these are the different types of neurons. They're unipolar. You can kind of see that for yourself. I don't need the pointer, but there's unipolar, bipolar, pseudo-unipolar, and multipolar. So I want you to, these are neurons, different types of neurons. I want you to look these up in the book and understand what they are. So this is the end of the first lecture in the course. I've tried to cover most of the content in Chapter 2, which discusses the cell types of the central nervous system. We will now move on to Chapter 3, discussing the neuronal membrane and what gives rise to the resting membrane potential. I've tried to cover, what's in the chapter. However, there's more detail in the chapter than I address in the slides. You'll be responsible for the chapter detail with respect to what I will ask you in the quizzes. So please do read over it very carefully.